All right, time enough for another lecture.
Okay, now we're going to talk about next generation genetically modified organisms. And specifically, this lecture is still about food. What is bioengineered food? In 2021, the US FDA started to require a bioengineering label on specific food products. That label might look something like this, where there's a circle and it says derived from bioengineering or bioengineered. There might also be something small on the back of your food products. Maybe right next to the nutrition facts could be a small statement that says something like contains a bioengineered food ingredient. Now, if you see these things on food in the grocery store, and I encourage you to check the next time you're there, or maybe to look in your own pantry and see, do you have foods that are derived from bioengineering? I want you to think, what does that mean? Right? Do you know what that means? According to the US FDA, a food that contains genetic material that has been modified through in vitro recombinant DNA techniques and for which the modification could not otherwise be obtained through conventional breeding or found in nature is a bioengineered food. Now, that's kind of a mouthful. And so, in this lecture, we're going to break down what that means. We're going to talk about all the different ways that food crops can be improved and what counts as bioengineering, at least according to the US FDA. Okay, so here's sort of the overview. Crops are genetically modified in many different ways. And we're going to go through each of them and talk about sort of the benefits, disadvantages, and the regulation related to each one. So the first crop improvement strategy that we're going to talk about is traditional breeding. This is crossing plants and then selecting offspring with desired traits. So if you have one plant that produces a lot of seeds and another that produces really big seeds, you might cross them together in order to get a plant that has a lot of really big seeds. This type of breeding approach is something that humans have been using for thousands of years. They've been observing crops with desirable phenotypes and crossing them together. And when they do this, you can affect just a few genes within the plant genome, or you can change entire genomes, right? You're remixing the genetic material of the plants as you cross them together. There's no safety testing required for this type of crop improvement, and there's no regulations. You know, this week we've been talking about directed evolution. Domestication, or the traditional breeding of crops to improve them, is essentially a very long directed evolution experiment. And through this type of approach, people have really changed the crops that we know and love today. These are ancestors of watermelons. You can see how much of the inside of the watermelon is actually rind and how much of it is just devoted to seeds. And humans in time have selected over and over again for watermelons with more of that like sweet pink flesh and fewer seeds and rinds to get the varieties that we have today. Same type of thing has happened for bananas. Here's a wild ancestor of banana. Again, is just chock full of seeds. And then maize, the wild ancestor of maize is actually something called teosinte, which has, much smaller and harder seeds than the maize that we grow as sweet corn today. So this cool kind of very long directed evolution experiment is still something that we use today in order to try to improve crop varieties. There are advanced methods for breeding today that allow you to kind of genotype plants when they are small, either as seeds or seedlings, in order to figure out if they maybe have the mutations from each parent that you're interested in. We sort of figured out for some traits which genes are important for them, and then can direct breeding through genetics instead of waiting for the whole plant to grow up. But the point really is that you can just cross plants together and get improved varieties over time. Now, another strategy is mutagenesis, another way to get a new crop variety or to change it. And in mutagenesis, you expose seeds to chemicals or radiation in order to make DNA damages, in order to acquire random changes in the genome. Chemicals and radiation can damage DNA, and then as your cells or the cells of the plant try to repair those breaks in DNA, they may not repair them perfectly, and you get changes in the genome. When you apply chemical or radiation mutagenesis, you usually affect hundreds to thousands of genes all at the same time. It's really difficult to just dial in the amount of radiation in order to get a single mutation in the backbone or in the genome of your plant. And often, actually, that's not that helpful because this is a random mutagenesis method. It would take a really long time if you were to apply low doses of radiation that change one DNA base at a time. Instead, you want to change a bunch of things all at the same time to increase the likelihood that you change a phenotype that you care about. So ruby grapefruits are actually an example of plants that have been developed using chemical mutagenesis. I think it was chemical, maybe it was radiation mutagenesis. Doesn't matter. Basically, farmers realized through one plant growing in a field that that grapefruits were able to produce this ruby color. There was one plant that acquired a mutation just serendipitously, and it made ruby red fruits. And the farmers who saw this thought that that would be a really valuable trait, that would make them stand apart from other grapefruit sellers in the market. And so they started applying mutagens to their grapefruit seeds in order to try to get more plants that also had this ruby phenotype. And so they were successful. There are actually, several different varieties of grapefruit that now all have this kind of really sweet and beautiful ruby red color. This type of mutagenesis does not require safety testing, and there's no regulation on it. So you may be eating lots of plants who have been developed using a mutagenesis strategy. Okay, there's a strategy called RNA interference. This is a method for turning off specific genes. The way it works is that you transform your plants, you introduce small RNAs into the plants that then bind to a gene of interest, to the RNA of a gene of interest, and then target that RNA for degradation. So double-stranded RNA inside of a plant cell is very unstable and often gets degraded really quickly. So if you introduce small RNAs that match a messenger RNA, then you can get it degraded. The example that I'm showing here is of a small RNA that targets virus RNA so that the plant cells are protected from a viral pathogen. That's actually because papaya were engineered to be resistant to a ring spot virus using this method. Almost all of the papaya that you eat is of the genetically modified variety. They are producing a small RNA that targets this ring spot virus and prevents it from making the whole plant sick. When you introduce RNAs like this into the plant genome, you can affect, 1 to tens of genes all at the same time. These short RNAs are short enough that it's possible that they would target several different genes in the plant genome, although often they are quite specific. There is safety testing required for this type of thing, and it's highly regulated. So this would count as a bioengineered food because there is no way of kind of naturally getting these short RNAs into plant cells. So they would count as a recombinant DNA technology. Okay, there's straight up transgenics. This is where you insert selected genes into the genome that are not RNAs. And so these small RNAs are sort of one class of transgenic plants. We separated them out just to be able to talk about the papaya ring spot virus example. But in practice, these two things are quite similar. The most famous examples of genetically modified plants being made with a recombinant DNA method are the Roundup ready or herbicide tolerant plants that have a single herbicide resistance gene overexpressed. And that makes the transgenic plants resistant to the herbicide. So you can spray this herbicide all over the field and your transgenic or your genetically modified plants will still grow. But weeds and other things that you don't want to be growing won't. The other really famous example are like BT cotton and BT corn. BT stands for Bacillus thuringiensis. It's actually a type of soil bacterium that usually produces a small toxin that is only toxic to a larval stage of insect or like a wormy type of insect. And when you put the gene encoding this toxin into the plant genome, then the plants can produce this short peptide that kills like these cotton bollworms or other types of pests and protects the plant essentially from these predators. And so these are nice because then you don't have to spray the plants with any type of pesticide. Instead, the plant is producing a little bit of this soil bacteria's genes to prevent predation from pests. Okay, so this usually affects, less than 10 genes, but it does require safety testing and is highly regulated because the US FDA, again, terms this type of modification of plants as a genetically modified organism. Okay, the last crop improvement strategy is gene editing. You can use proteins like CRISPR-Cas proteins in order to make really targeted deletions from the genome. The way CRISPR works is it's essentially a protein that likes to cut DNA that is targeted to specific DNA sequences using a short guide RNA. And when that guide RNA finds a region of the genome that it matches, then the Cas protein, Cas9 in this case, sits down and cuts the DNA there. After a cut is made, the cell usually tries to repair the DNA, but will make deletions in trying to repair it. So this all is an example in tomato where the authors were trying to increase the amount of vitamin D that the tomato produced by deleting an enzyme in a biosynthetic pathway. Then in this case, they're deleting this DR2 enzyme. And by deleting this part of the pathway, they can increase flux through the pathway towards vitamin D. So interestingly, this type of deletion, even though it's highly designed, you do transform in, or you deliver in this Cas9 protein and these guide RNAs. Because you're only deleting portions of the genome, this is not regulated, at least in the United States. So there's no safety testing required and there's no regulation of it here. In Europe, it's a different story. This type of modification would still count as a genetically modified organism, and there these would all be prohibited from sale. So sort of interesting. There's a spectrum of methods by which you can modify plants, and those methods are all regulated somewhat differently. Now, how do you get DNA into plants? This is a question that I would have had if I were in your shoes. Like, okay, that sounds interesting, but like, how do you actually get DNA into plants? The answer to this question actually lies at the center of this crown gall tumor. Inside of this kind of weird plant tumor is actually a little bacterium. It's a bacteria called Agrobacterium tumefaciens. And this bacteria is kind of amazing. It actually takes a portion of its genome and pumps it into the plant cell. So this kind of red region down here, gets copied and then moved into the plant cell. The bacteria actually makes this little protein-like needle to poke a plant cell. And then through the middle of this needle, they pump in DNA and they pump in some proteins. These proteins help shuttle the DNA into the nucleus. And then once the DNA from the bacteria gets into the nucleus, it actually integrates itself into the plant's genome. Now, Agrobacterium usually delivers instructions to the plant cell to deprogram it. So it will tell it, okay, stop being a bark cell, for example, stop being a, vascular cell inside of the middle of a plant stem. Instead, you should just turn into a sugar factory. Make me a lot of sugar. And the plant cell doesn't necessarily know that this DNA came from a bacterium, and so it will start expressing the genes encoded on that DNA, and it will turn itself into a sugar factory that replicates over and over and over again and forms this big kind of tumor-like structure. So in the middle here, you can see all of these like very clever, very happy bacteria eating lots of plant sugars. Okay, but you can actually use agrobacterium to deliver DNA into plant cells in order to make transgenic plants. So we can take that little segment of DNA that usually gets transferred and we can recode it. We can actually remove the genes that tell the plant to become a little sugar producing factory, and we can put in instructions for other things like produce a fluorescent protein or, produce a BT toxin, something like that. One of the easiest ways to actually get transgenic plants is to take the plants and then dip the flowers into a solution of agrobacterium. In this solution of agrobacterium, the bacteria will donate DNA or conjugate DNA into the egg cells within the flower. Those egg cells will then get fertilized by pollen and become seeds, develop into seeds. And some of those seeds will then be transgenic. They'll then carry the what's called tDNA or transferred DNA that was given to it by the bacteria. So here's just examples of what the seeds of this plant, which is Arabidopsis thaliana, look like under white light. And then you can see we delivered into them this red fluorescent protein, and the seeds then glow when they have that red fluorescent protein. Okay, but floral dip, this method does not work on all plants. Arabidopsis thaliana, this model plant, makes tens of thousands of seeds in just like a single pot of plants. But something like tomato makes maybe hundreds of seeds, maybe less per plant. And so if you're not able to deliver DNA efficiently into each egg cell, then the likelihood you would get transformed seeds is pretty low. There's also issues with just like the shape of the flower and the access that the bacteria have to the egg cells within the flower. And so it seems to not work in every plant variety. So what do you do? How do you make a transgenic tomato if you can't just dip the tomato flowers into a solution of bacteria? Well, you can take advantage of the fact that plant cells can usually de-differentiate into what's called callus. So here's a little piece of a leaf that has been treated with hormones that cause some of the leaf cells to de-differentiate into callus. So this callus are basically like undifferentiated plant cells. You can think of them sort of like plant stem cells, which you can then treat with a different series of hormones to get this callus to redifferentiate into leaves and then redifferentiate or produce roots. And so you can go from callus undifferentiated cells into whole plants. Now, why is that useful? Well, you can take callus and you can incubate it with agrobacterium. And the agrobacterium will again conjugate DNA into the plant cells that it comes into contact with. Then you can take that callus and you can put it onto selective plates. So here, if the agrobacterium is delivering DNA that encodes for your genes of interest, maybe your fluorescent proteins, maybe something else, it may also encode for an herbicide resistance gene or another selectable marker. And you can put the callus onto plates that contain the herbicide, and only the ones that are transgenic, only the calli that have received DNA from the agrobacterium will continue to grow. Everything else will turn sort of black and die. But those ones that grew, you know that they contain DNA from the agrobacterium. You can then take that callus and you can put it onto new media plates, onto other plant hormones to start to get shoots and then roots to form, and you can actually regenerate a whole plant. So that's the process by which people make transgenic plants. You can, instead of using agrobacterium, use something called a gene gun to inject little gold particles that are covered in DNA into the cells of the plant, and then do the same type of selection and regeneration methods. It's not as popular because these gene guns are quite expensive, and usually you end up with many, many copies of your transgenes inside each plant cell. And so people don't like that because you have a little bit less control using a gene gun than you do with the agrobacterium. You can also deliver DNA into plants using carbon nanotubes. So there's like a little tiny tube of carbon that you can attach DNA onto and then deliver into plant cells just using a very little amount of force. You can get these things to permeate plant cells, and then the cells will express fluorescent protein. So here's GFP in 4 different plant varieties after having it delivered with these little carbon nanotubes. So that's kind of fun and exciting. One of my favorite methods for producing transgenic plants is really new. I would say it's not all that widely used yet, but it's an exciting idea that you might be able to get plant transformation without having to go through this regeneration step. Because it's actually at this step where the transformation of many plants fails. You may be able to get callus that's resistant, showing that you got your DNA into the plant cells, but you might not be able to get new shoots or roots to form because you don't have exactly the right conditions, and then you can never make these transgenic plants. So if you can avoid that regeneration step, then you might be able to make transgenic plants of different varieties, or at the very least, you wouldn't require so much specialized equipment to put the callus into exactly the right conditions in order to get the transgenic plants you want. Okay, so sorry, this is a picture of a plant's meristem. This is the portion of the plant that is actively growing. It is the part of it that will produce flowers and seeds. If you are to cut it off, the plant would determine a different location to have a new meristem grow. It will basically not like that you have cut it off and it will try again. If you prune things, this is often what you're doing. You're cutting off this meristem. Now, there were some authors, some scientists at the University of Minnesota who figured out that if you cut off the meristem, then deliver agrobacterium, which conjugates DNA into the cells, along with a couple of developmental regulators, some proteins that stimulate meristem development, you can actually get a new meristem to form that contains your edited genomic DNA. So here the authors were they delivered DNA encoding Cas9, that CRISPR-Cas system, targeting phytoene desaturase, which is an enzyme that's important for chlorophyll development. And if you knock it out, you get these really white plants. And so what they showed is that, yeah, you can cut off the meristem and then new shoots will form that are edited, and you can collect seeds from them, and those seeds will be edited, and you can make new modified plants without having to go through the tissue culture. So I thought this was really fun, and I wanted to share with you how that was done. But where is all of this leading? I promised you that we would talk about the next generation of bioengineered or GMO plants. And what I think is that these plants will have more precise control over gene expression or more nuanced phenotypes. So the first generation of GMO plants had single genes expressed through the entire plant. So this might be that Bt gene expressed in every cell within the plant in order to make the entire plant resistant to the insects that were trying to eat it, or it might be the herbicide resistance gene expressed in every cell within the plant in order to get the whole plant resistant to that herbicide. And that's great for features like pest resistance or herbicide tolerance. But I think the next generation of plants is going to have genetic programs introduced to them to control gene expression in very specific cell types at very specific times in development. And I think that people are going to be doing this in order to make stress resilient crop varieties. The new challenge is really to see if we can grow crops in increasingly hostile climates, and being able to tune the activity or change the activity of specific parts of the plant can be really important for that. You don't need the whole plant to start, like, okay. So if there's drought, for example, you might want the root system to elongate to start getting deeper. And that might allow it to reach water in layers of soil that have not yet dried out. But you want that only to happen in the root system. So the plant is devoting resources to its root system to get it to elongate. If you have that elongation program running, for example, in the above ground portion of the plant too, now you have two things that are fighting each other, right? The shoot system's trying to grow and the root system's trying to grow. And maybe there won't be enough resources for both to be able to do that. And you'll never reach the water in those deeper layers of soil during the drought. So it would be great to be able to control gene expression in specific portions of the plant to really do some kind of fine tuning of plant resilience. And we're starting to get the capacity to be able to do this. This is an example from my lab just showing you how precise control over spatial patterns of gene expression across a plant can be important for engineering specific features of a plant. So we're looking at our 3 different Arabidopsis plants. Arabidopsis is our model system. It's a really small and easy to work with plant, and it's easy to transform. A wild type or unmodified Arabidopsis has a long primary root that grows down in the direction of gravity. All these root branches that also grow down in the direction of gravity. And if you were to zoom in on this root system, you'd see that it's covered in these tiny root hairs, which are really important for water and nutrient acquisition. Now, if you express a mutant developmental regulator from a promoter that's active in lots of different cell types throughout the plant, you can change the development of the plant pretty severely. So this engineered plant has no lateral roots. It also has a shorter primary root. That primary root has a hard time sensing gravity, which you can tell because it no longer grows straight down. It doesn't have any root hairs, and it's got several structural changes in the shoot as well. And one of the reasons that there are all these different impacts is because this gene is expressed in all these different tissues throughout the plant. Now, if we take this same gene and we only express it in one cell type, in these what's called xylem pole paracycle cells, these specific cell types within the plant root system, then we can make a targeted change in the plant's structure. So here, the plant does not produce any lateral roots, but it's otherwise identical to the wild type plant, right? So with this precise control over gene expression, we can make a precise change in the structure of the plant. We can actually take it one step farther than that and tune the number of root branches that the plants make. So with different synthetic genetic systems, with different gene expression levels, essentially, in a tissue-specific manner, we can control the density of root branches. And now we're wondering, okay, can we tune root structure for optimal growth in a specific environment? So we're not the only people by any stretch who work on this type of precise plant genetic engineering, and we're excited to see where it goes. After this lesson, I'd like you to think about the FDA's definition of a bioengineered plant or a genetically modified organism. Do you think that they're drawing the line in the right place? Do you think it makes sense to have regulation that's based on the process by which you have developed an improved crop variety? Or is there some other metric that you would prefer to see labels on or about on your food in the grocery store? Do you look for GMO or non-GMO labels when purchasing food? Why or why not? Did anything surprise you about the way transgenic plants are created? And then, would you feel differently about GMO plants created to withstand climate change versus those engineered to produce pesticide? Or are those both the same to you? Is a GMO always a GMO? Or does it matter kind of what the purpose was for producing that plant? I would love to know what you think of these GMOs. And if you feel like sharing your answer to these questions with me, I'd love to get an email from you. Hope you enjoy the rest of the week. Bye.
Wow. Okay, that's very interesting. Just trying to remember what all these things that they mentioned are. Oh. Well, maybe I'm keeping up. I don't know for sure.